Merhabalar. Yıldız Teknik Üniversitesi Biyoteknoloji ve Genetik Kulübü Biyogen olarak hazırladığımız, bu sene üçüncüsünü düzenlediğimiz Astrobiology Konferansı'na hoş geldiniz. Hepinizi aramızda görmekten büyük mutluluk duyuyoruz. Etkinliğimize başlamadan önce sizlere yapacağımız çekilişlerden bahsetmek isteriz. Parla Candle desteğinde gerçekleştirdiğimiz mum çekilişine katılmak için katılım formu oturum aralarında sohbet kısmında paylaşılacaktır. Ve YTÜ Biyogen Instagram hesabımızda paylaşılan çekiliş şartlarını yerine getirmeyi unutmayınız. Ek olarak oturum arasında oynayacağımız Kaot oyunu ile YTÜ Biyogen Instagram sayfamıza duyurulan ödül kitabımızı kazanabilirsiniz. Kaot zamanını takip etmeyi ve katılmayı unutmayınız. Şimdiden bol şans dileriz. Şimdi sizlere günün programını özetlemek isterim. Konferansımız bugün toplam 3 oturum olmak üzere saat 12.00'dan 15.45'e kadar sürecektir. Oturumlar arası 45'er dakika molamız olacaktır. Etkinliğimize dair katılım belgesini almaya hak kazanabilmek için oturum sonlarında paylaşılan ve aralarda geçen QR kodları ve link bağlantılarından oturuma katıldığınıza dair formları doldurabilirsiniz. Katılım belgesi alabilmeniz için en az 6 oturuma katılmanız gerekmektedir.
Esteemed participants, greetings. Welcome to the Astrobiology Conference organized by the Biotechnology and Genetics Club, Biyogen, of Yıldız Technical University. We are delighted to extend a warm welcome to each one of you. Allow me to present a concise overview of today's program. The conference will unfold across 3 sessions, spanning from 12 o'clock to 3:30 with 45-minute intermissions between each. To be eligible for the participation certificate, we encourage you to complete the forms shared at the conclusion of each session, utilizing the QR codes and links provided during breaks. A minimum attendance of 6 sessions is requisite for the certification.
Bugünün ilk konuşmacısı olan Sayın Arda Deniz Dokuzoğlu'nun sentetik biyoloji ve yaşamın ortaya çıkışına termodinamik bir bakış açısı konulu sunumunu yapması için davet ediyorum.
I now extend an invitation to Mr. Arda Deniz Dokuzoğlu, our inaugural speaker, to deliver his presentation on synthetic biology and the thermodynamic perspective on the origin of life.
So I start, right?
Yes.
Okay. Let me share my screen. Okay, it should be there. My name is Arda. I'm actually an entrepreneur rather than a scientist, more of an independent researcher maybe. I have a company called Gene. We work on synthetic viruses, synthetic biology, mostly on therapeutics. And today I will present you more about what we did before throughout our R&D projects, let me say, and especially today will be concerning the project that we accidentally synthesized the world's first viroid, synthetic man-made viroid. It is actually completely designed in a computer and synthesized chemically and booted up in a bacterium. And I will also mention you how this got me interested in the thermodynamics of life. So thanks for giving this chance for me to present, and let me start. Let's have a brief look in what constitutes a cell. These are the major molecules inside the cell that we're concerned investigating these technologies. We have DNA, RNA, proteins, and metabolites, and these are more or less make up the whole system. I won't go much into detail as these are very general information, but there is one specific thing we need to point out. These cells are not just in production facilities. They don't just produce enzymes, metabolites, proteins, and structural components. Cells, as a form of life, also have the capability to process information. And they do it by genetic regulatory networks. So they actually respond to stimuli by pre-programmed biological processes. And you can like one example for that is the development of the embryo. Here you can see some native genetic regulatory networks and how they control this very sophisticated process of development. My engineering field is more interested in developing artificial genetic circuits. So designed circuits that actually programmed to respond to stimuli like chemicals, light, heat, antigens, any fluctuations in the surroundings, and calculate the response, like decide on a response and then execute it, actuate it. And this more looks like developing an electric circuit. And there are like AND gates, OR gates you can do inside a cell. So this is pretty much developed field now. This is some examples what you can do with genetic circuitry. I will pay attention to the lower left corner, the repressilators, because we have an interesting story about the repressilator. But yeah, the problem with genetic circuitry is the inside of a cell is a very very packed, very complicated, very noisy environment with nanometers of gaps only between the molecules. So there is actually a quite good isolation between them, because proteins generally know where they're going to bind and actuate, but still there are some problems with it. It's a very complex system, a lot of genes, a lot of genetic regulatory networks, and a lot of cross-talking metabolic cycles. We can summarize them in 6 main causes of problems, and these are like unknown parts, incorrect kinetic parameters, crosstalk between genes, some extrinsic noise, intrinsic noise, which arises from the internal process, and then a metabolic burden. When you put a genetic circuit inside a cell, it generally steals the resources that are required for the natural growth of the host organism. And we decided to fix this problem by creating a separated central dogma, separated genetic language, let me say. And this this actually resembles a virtual machine in a computer. So what we do is we separate the language, main language, from the host cell. They still share the same resources inside the cell, say the energy sources, even ribosomes that are required to translate proteins. And in this quest, we try to develop an independent replication mechanism, an independent transcription mechanism, and semi-independent translation mechanism. And in the end, we ended up with a virus without a capsid, which is a viroid. And this was really unintentional. This is the final product, but let me go into the details of how we actually developed these synthetic life form. And we started with replication. It was a double-stranded DNA. We chose a double-stranded DNA because it was easy. Actually, it was the only way back then to have a artificial replication mechanism. It used 5 proteins, which includes DNA polymerases, helicases, cap proteins, priming proteins. So 5 of it's quite simple mechanism, and it is actually borrowed from bacteriophages. And with 5 proteins, we were able to effectively replicate this piece of DNA. Of course, there was the need for transcription, and we used a very standard method for that, and we incorporated T7 RNA polymerase. And for the translation, we actually got inspired by Stanford, like one lab from Stanford. And at that time, they suggested these orthogonal mRNAs, which means and specifically encoded mRNAs that are programmed to steal some of the ribosomes of the cell, not all, but some of the ribosomes. And we do that by leveraging the key and lock relationship between the ribosome and the 16S rRNA. We reprogrammed the 16S rRNA, and then on our mRNAs we had a matching specific binding site. So this way we separated the ribosomes, which process the native proteins and the proteins that are synthesized on our plasmid, which is actually a viroid. Overall, this was a good approach, but in practice it failed because it was too toxic to the host bacteria. And the reason for that is it just replicated without control. It replicated rapidly and started to kill cells. So at that point, the solution we came up with was actually again an inspiration from a bacteriophage lambda. And we decided to use these genetic circuit inside bacteriophage lambda that helps it recognize how many copies of viruses are inside a bacterium at a certain time. And we needed a little bit of more detail to that. So this is like a two protein system in the native form. With its simplest form, we needed a three protein system, and the reason for that is we need 3 different states: a state where we encourage replication, a state where we maintain the current rate, and a state that we actually stop the replication. So we needed 3 different states, and for that we actually used a repressilator. A repressilator is what you see here, is like three genes, let's say gene A, B, and C, blue, green, and red. And we constituted this relationship where every protein represses the other 2 at a specific rate. And there is a very specific distribution of KD values inside this system, where you get a calculator, an analogous calculator that counts the number of DNA molecules, which is the DNA molecule that codes these proteins, codes these genes. First we go to the computer simulation. We run Monte Carlo simulations and use brute force actually in order to get the exact KD values we need to constitute between these repression cycles. And you can see here on the left, these are the KD values specifically needs to be integrated, needs to be built. And for building them, we went to a very innovative direction, and we used DNA repressing proteins, a protein called TALEN. You probably heard of it. So it's like a gene editing protein, and we used the Gibbs free energy calculations to engineer the binding of the DNA to the TALEN proteins with this very specific KD value. And this is how we went. And working on this, I actually realized like the whole thing that we simulate worked on this Gibbs free energy calculations. Think about every relationship as a chemical reaction. Simply what we end up in the end is a very complicated chain reaction with a very specific mathematical model. And to develop that model, actually, we used a program called Tinkercell. Tinkercell was I think it's like a PhD project of some guy, but then they stopped updating it, stopped improving the program. But I think it was one of the best solutions out there back then, and there were some bugs inside the algorithm, so we went in and tried to fix it ourselves. And we were successful to a point where it met our needs. So yeah, we actually first deterministically modeled every relationship inside the viroid and like the whole genetic circuit. And also we went a little further and did the stochastic simulation, which also again supported by Tinkercell. I actually want to show you how it looks like. I just have a very brief version here. I want to show you this because I want you to get encouraged to get this software and develop it further, because we need it. So yeah, this is like you choose your parts here. You can put a coding part. You can choose an RBS, which is important for translation speed. A promoter, which promoter with activators and repressors, which gives you the rate of transcription. And it's so well integrated. You can actually like tap onto this one and see the mathematical model right away. And you can actually build a very complicated genetic circuit here and simulate it quite easily. You just need some well-maintained algorithm for this program, which is on you because nobody's developing it anymore, which is a pity, I think. Yeah, so basically the project actually funding dried up at this point. We couldn't test this this calculator, which we really wanted to do. I tried to continue the project in in the US, I tried to collaborate with biohacker spaces, academicians, some independent researchers. Nobody was interested that much, so we had to stop this project at this point, and we moved on to another therapeutic project. But the importance of this project, or genetic circuitry in general, for regarding astrobiology, is that this is a very effective engineering method for you to, let's say, engineer a bacteria and to send it to let's say Mars or another place, to terraform maybe, because in that case you need a very specific organism, very resistant or adapted to a specific environment, and you don't have like many chances. Like you cannot send tons of lab equipment and build things there, but it is easier to build cells here, engineer cells, put some genetic circuits, well-tested genetic circuits inside the cell, and send the cell to the destination, and let the cell get the work done. So it's like a very compact, self-replicating, self-expanding engineering project. So I find it very relevant in the field of astrobiology. Anyway, working on these projects, on the other hand, got me interested in the mathematics of biology. So how does the biology work in terms of mechanics? How it have emerged, how the biology knows what to do, right? And that got me into thermodynamics, and then for sure in entropy, and that got me thinking, actually we're in some sort of illusion on Earth. It is a very specific place in the universe where we see constant progress. We're used to constant progress. We're used to things getting sophisticated, things getting improved, which is not the case in anywhere in the universe. I mean, like it's pretty rare. And to explain that, I need to go into the second law of thermodynamics. Think of a glass, and glass itself is a very sophisticated form. It's like produced by using knowledge and energy, and there are millions of ways of a glass to shatter, but there are so few ways to build it back from the ashes. That is because there is this intrinsic property of universe that tends to destroy things. It tends to bring more disorder. And to explain that, I need to tell you more about the concept, the notion of entropy. And then entropy is a measure of disorder in a system. You can also say that entropy is how much we don't know about a system. There is also the informatics way of explaining it, which is entropy is the informational value of a system. There is another explanation in quantum mechanics, which is a version of Gibbs entropy, which is how much we don't know about a system. I showed you this equation, delta G equals to delta H minus T delta S. Delta S there is the change of entropy. And if you want to think about it, you can think about bricks. If you put bricks randomly, you don't expect them to pile up in an ordered fashion. They rather will be all over the place, right? that's because it is very unlikely to have a randomly scattered brick will pile up in an ordered way. So it's much less likely. So as things get more ordered, they have lower entropy, and they have low randomness. They're more predictable, and there are less ways of configuring that system. And as it goes higher in disorder, there are much more ways to reorganize and configure. Another example is, like, think about a gas chamber. You have a separator in between, and on one side there is a high temperature gas, and on the other side there's low temperature, and you expect a heat transfer between them. They're not in thermal equilibrium, so they're low in entropy, because it is rare to see such thing, right? When they free up, they will eventually come to thermal equilibrium and even out all around the chamber. This is the formula. Why it's important? Because entropy has this logarithmic nature, so it slowly progresses to the maximum. So why is it important? Because it is actually a good way of explaining why life has emerged on Earth. But for that, we need to define life. NASA has a definition: life is a self-sustaining chemical system capable of Darwinian evolution. I think it's not enough. It's not. It misses some points. A better explanation is life is a self-sufficient chemical system, far from equilibrium, which means it's low in entropy, capable of processing, transforming, and accumulating information acquired from the environment, which is a very important part of it. And actually, I will connect Darwinian evolution with this part of acquiring information in the following slides. There are different ways of approaching defining life. Some even don't define it and just talk about its properties, its features. So let's go that way and ask, what is life? What makes biology, different than chemistry? And at that point, we can talk about it's pre-programmed. It has a memory. It has some information. It contains some information. Another thing is it adapts. It learns. It evolves. Third one is it is ordered. It's regulated and ordered. The fourth one is it's reproducing. It inherits information, and it grows. It acquires more matter in its organization. So it actually acquires the matter, and organization is another one. Very, very important property. Life in general has boundaries and compartments, so it's very well organized system. Homeostasis is one property, but homeostasis is also maintained in sea, in the ocean. So it's not very specific, but living systems have homeostasis. They have a metabolism. They respond to stimuli, and they process energy. These are like less specific ones. And a very good other question is, why doesn't life occur elsewhere? So why on Earth, right? We will answer this a little later. But we go back to how life emerged, and what could be the medium for boundaries before the lipid bilayers. So there are rock microcavities, clay particles, some solid colloidosomes, aerosols, gas bubbles, ice crystals, between the gaps between ice crystals. So these are thought or hypothesized that might have been the early mediums of early sources of boundaries. And you need the boundaries because you actually want to retain the information of life inside somewhere, right? And going back to entropy, one of the very specific features is that life actually retains low entropy. If think about the gas chamber, life needs to set a boundary and retain the low entropy in order to function, to evolve, to have more and more organization inside, to grow and to function. And a recent, relatively recent finding is that this is actually hypothesized much before the vents beneath the ocean were discovered, this thermal vents. It is actually hypothesized that these thermal gradients inside on the depth of the ocean can be a very efficient place to have life formed, because it actually also works as a catalyzer for conversion of organic molecules, and it again retains these molecules in small volumes so that they can actually find, so that the energy provided to them is enough for converting into more complex forms. And when you look at the hierarchy of complexity in the biosphere, you have, starting from the subatomic level, you have more organized atoms, molecules, organelles, cells, and then organisms, communities, ecosystems, and all the way up to the biosphere. And it's actually a hierarchy of complexity, and we cannot actually say these are separated from each other. And when we look at the biological development process, we can say as events roll out, which means actually as time progresses, the biological components get more complex, they got more ordered, they got more sophisticated, they moved further away from equilibrium, and they get more specifically configured, which is actually an act of entropy. So they actually acquired lower entropy, a state of lower entropy, by actually expelling high entropy outside. What happens when you put two molecules side to side? So let's say a protein and a small molecule, you put them side to side. What they do is they generally tend to fall to the least energetic state that they can access. There might be more less energetic states, but they might not be accessible. So they stabilize by decreasing their free energy. So going back to the equation, that actually helped us engineering all these genetic circuits, all these protein DNA relationships. It is actually the natural property of universe that these molecules tend to get into a lower energy state. And by lower energy state, they can do this either by losing heat or increasing entropy, which is the natural thing. And when you look at let's say this is a enzyme catalyzation graph. You can see there is this free energy loss in state A and state B. And between state A and state B, there is this transition state which requires activation energy. The interesting thing life does is that enzymes actually manipulate this landscape. I can actually show you the landscape here. It looks like this. So this is the vertical axis is the free energy, and the lower tipping points are actually the stabilizing stable points, the low energy points, local minimums for a protein to fold. So this is a protein folding graph. And you can say the 2-dimensional plane is actually stands for the entropy that the protein has. As it loses energy, it also loses entropy. So it gets into a lower entropy, but this is counterintuitive because these proteins are actually increasing the entropy of the solvent, which is water around them. So the net entropy actually goes up, and they fall into a lower energy state. I don't know if you see my pointer, like the cursor, but I will point anyway. So let's say an unfolded protein naturally goes down to this well in the middle, which is a low energy state, and it doesn't have enough energy to discover even lower states because there is an activation energy between them. So enzymes, in this case chaperones, help them jump from this obstruction and go to the maybe more functional state. And this is very interesting, actually. This is very, very interesting because this is why I call the Earth a matrix. Because an enzyme, what the enzyme really does is actually it bends the reality. It bends the free energy distribution. It changes the landscape, and it does this by having the information, having the blueprint of how to do it, or of how to convert a molecule from form A to form B. And this is very important. Why? Because I will show you how entropy and information are actually linked. So this is Maxwell's demon. It is again a gas chamber, and this actually puzzled physicists for 150 years probably. The idea is, in a normal setup, you would expect a thermal equilibrium to stand as is, like to continue being in the thermal equilibrium, because it's in the highest entropy state, and it cannot go in a less entropy state naturally, right? But Maxwell comes up with this idea that there is this demon who knows the position of every molecule, and it allows that by just controlling a little door between these cells, he just lets the hot gases, like the fast moving molecules, to one side, and slow moving molecules the other side, just by knowing which is fast and which is slow. And it actually looks like it violates the thermodynamics, like the second law of thermodynamics. And very recently, I mean, like 1980s, it was quite probably that we figured out that actually what happens here is that, like, because this actually this demon made this system a lower entropy system, right? Without increasing entropy elsewhere, we thought. But it turns out that in the mind of the demon, there has to be a record-keeping system. There has to be a memory, and recording something to the memory or erasing it actually increases entropy. So that's where the entropy went. This is actually the first time that information got its connection with entropy, and now we know that low entropy actually comes with complexity. The bond is still not very clear. There's still progress to do there. But yeah, we know that entropy increase by destruction of information, and it is a quantum property, and information is a physical entity. Why is that important? Because we will move to evolution, and evolution is a natural method of learning, and it seeks harmony. And what evolution does is actually learn, which is create information. And we define life with an attribute of learning, which means life is actually acquiring information using evolution, which is actually a process of obtaining low entropy. So this bond makes evolution an emergent phenomena. We can say existence in the form of organized matter is the memory where biological information is stored. This is like a hypothetical claim, let's say, I'm doing right now. But let's go into the details of evolution. And before that, let's coin that evolution is a population reaction. It tries to make harmony with the environment, so it has to somehow communicate with the environment, and that communication is the evolution cycle. Naturally, a biological information degrades over time, let's say mutations, and most probably most of these mutations are detrimental, but some of them by chance are actually more harmonious with the environment. And when these traits go out and function with the environment, communicate with the environment, and as a result, only the harmonious traits get selected, what happens at this exact point after selection is that by the existence, the biological information now is recorded in the biosphere by reorganizing the matter in its own information. This is actually a way of manipulating entropy, and this makes me think, what happens if we destroy it? So how do we destroy information? Like what we see in nature is the information is destroyed by mutations, by mass extinctions, by death, and also selection itself is a destruction mechanism. It actually erases the information, and this takes us back to the Maxwell's demon. Actually, naturally, as a natural response, it goes to disorder. There's destruction, where we also emit entropy. But every once in a while, some of these destructed codes, they're able to survive, and they get recorded in the memory. And after a while, you think, won't you think, the Earth actually contains a lot of complexity by this way. Like it acquires more and more information, it develops more complexity, and continuously destroys it. And even let's go further into the future, there will be an asteroid maybe hitting the Earth, and the whole memory of biosphere will be demolished. And it is actually an act of entropy. It's like increasing. It's a good way of increasing entropy. Let's take it this way. When you look up to the sky, there are stars, right? And the reason you are able to see them is that the photon from the star actually traveled millions of light years away to you, to your eye, intact, so you can see them. So it's actually very difficult to destroy a low entropy photon, which is, let's say in this case, a visible photon. But for every photon, visible light photon that hits the Earth, we actually emit around 20 infrared photons. They're actually equal in energy. They have similar. There is a balance between the energy coming in and going out. But what's changing is we're actually sending the same amount of energy in a higher entropy format. So one way of Earth feeding on entropy is photons from the sun, and the other is from the beginning, as a part of the sun, we still have inside the core of the planet, we have a lot of free energy, which means ordered energy. We have useful energy. So these two mechanisms, the photons by photosynthesis, which is actually an invention on the way, but on the other hand, there is still free energy inside the core, which might have actually gave the source for life to emerge in the depths of ocean as a ever available form of free energy. And if you feed low entropy to a system for long enough, now we know that it has to acquire more and more information, which actually pushes the system, the chemical reaction, to get more and more complex, more and more ordered, more and more sophisticated. And I cannot say this is like a cause or a result, but these are very closely connected phenomena. And as a learning mechanism, evolution actually is part of it. Learning, neurological learning is also a part of it. Cells developing organizations, organoids, some genetic circuits inside is actually part of this process. This is more or less what I want to say. I don't want to, take too much time. I'm probably over a little bit. Maybe we can skip to the Q&A. What do you think?
Uygun hocam. Birkaç soru geldi bana da.
Türkçe?
Türkçe sorular.
Tamam.
İlkinden başlıyorum o zaman.
Hı hı.
Bir arkadaşımız şöyle demiş. Yaşamın ortaya çıkışında tartışılan bir yapı olan misallerin belirli bir konsantrasyona ulaştıktan sonra misal olduğunu okumuştum. Yani parça parça bir araya gelmektense bir anda oluşuyorlar. Doğruluğu var mıdır? Bu termodinamik açıdan nasıl mümkün olabiliyor? Buna benzer başka yapılar var mıdır?
Tabii. Aslında mesela lipid bir layer'larda, yani hücrenin mevcut yapısı sadece misaller değil. Hatta mesela self-assembly yapan proteinlerin çoğu da, yani kendi kendine organize olan protein, slide açık önümüzde. Eğer iki olay olarak düşünelim. A olayı, B olayı. Mesela miselin dağınık hali, miseldeki fosfolipidler olsun, fosfolipidlerin dağınık hali ve misellenmiş halleri. Bunların kendi kendine misel haline gelmeleri aslında daha düşük bir serbest enerji haline gelmeleri demek. Bunun sebeplerinden bir tanesi de suyla olan hidrofilik hidrofobik etkileşimler. Protein katlanmasında da serbest enerjinin %90'ı bu hidrofilik hidrofobik etkileşimlerden kaynaklanır. Yani proteinin o hidrofobik kısmı içine çöker kendiliğinden ve o enerjinin %90'ını azaltır. Hatta şurada bir örneği var. Yani bu proteinler mesela unfolded haldeyken entropileri yüksek, şeyleri yüksek, serbest enerjileri yüksek. Serbest enerjisi düşeceği bir noktaya gitmeye çalışıyor. Bu da istatistiksel bir kavram bu arada. Yani niye serbest enerjisi düşmek istiyor diyoruz? Çünkü serbest enerjisinin düşmesi için milyonlarca yol varsa serbest enerjisinin yükselmesi için çok bir iki yol var. Yani aradaki istatistiksel orantısızlık sebebiyle meydana geliyor. Yani bu molekülün düşük serbest enerjili formda durma ihtimali çok daha yüksek olduğu için bu forma geçiyor. Misallerde de bu aynı şekilde serbest enerjisi azaldığı için o forma geçmeyi tercih ediyor. Yalnız bazı spesifik kondisyonlarda bu oluyor. Neden böyle? Burada formüle bakarsanız delta S'in yanındaki T'nin önemini aslında anlayacağız. Sıcaklığın ne olduğu, entropinin ne kadar önemli bir faktör olduğunu etkiliyor. Şurada bahsetmiştim. Serbest enerji kaybederken protein aslında entropi de kaybediyor. Ve suya da bir miktar entropi şey yapıyor. Ama bu bir trade-off. Sıcaklık yükseldiği zaman bu proteinin daha yukarı bir noktada stabilize olmasını bekliyoruz. Çünkü entropinin etkisi artıyor equation'daki. O yüzden de mesela atıyorum sıcaklık 40 dereceyken misal oluşamıyor. Ya da 50 dereceyken misal oluşamıyor. Çünkü şeyi yetmiyor. Sıcaklık katsayısı olduğu için entropi ile entalpi birbirini dengeleyemediği için o state'deki Gibbs enerjisi daha yüksek bir yerde kalıyor ve oraya varamıyor. Genel olarak böyle anlatabiliriz. Bu aynı zamanda şunun da anlatımı. Mesela serbest enerjinin dünyadaki serbest enerjiden, dünyaya düşen serbest enerjiden çok daha fazla serbest enerji güneşin üzerinde var. Niye yaşam güneşin üzerinde gelişmiyor? Çünkü güneşin üzerinde sıcaklık bu karbon kimyasının sahip olduğu konfigürasyonları mümkün kılacak düzeyde değil. Gibbs serbest enerjisi farkı hiçbir zaman negatife düşmüyor o alan için. Dolayısıyla da orada yaşam gelişemiyor. Aslında şundan da bahsetmek lazım. Dünyada yaşamın gelişebilmesinin en önemli sebeplerinden bir tanesi sweet spot diyebileceğimiz bir noktada olması. Ne için? Karbon kimyasının sunduğu konfigürasyonların hepsinin bir var olabileceği bir aralıkta bir şey sunuyor. Dünya şartları, sıcaklık, ortalama sıcaklık ve var olan malzemeler, bir de solvent tabii su. Suyun varlığı aynı zamanda bu entropinin, yani dissipation of energy için de lazım. Yani o protein katlanırken bir şeye vermesi lazım o entropiyi. Mesela suyun aynı zamanda böyle bir görevi de var. Yani suyun varlığının bu tip reaksiyonların olabilmesi için böyle bir etkisi de var. Dolayısıyla o her yerde gelişemiyor. Olası konfigürasyonlar ne kadar fazlaysa maksimum entropi de o kadar yüksek olduğu için o ortamda gelişebilecek kompleksite de aynı oranda yüksek oluyor. Çünkü o kadar konfigürasyonun arasından daha spesifik bir, yani şöyle bir konfigürasyonun gerçekleşmesi, olası konfigürasyonların sayısı arttığında, olası konfigürasyonların sayısı olasılığı düştüğü için değeri de artıyor. Böyle de bakmak mümkün.
Başka bir soruya geçeyim. Yaşam ortaya çıkarken, özellikle yapıların düzenli hale geçerken termodinamiğe karşı gelen durumlar meydana gelmiş midir? Ya da karşı gelen durumlar olabileceğini tahmin ettiğiniz durumlar var mıdır?
Şöyle, lokal olarak zaten belli bir olayın, belli bir kimyasal reaksiyonun reversible olmaması için bir sebep yok. Yani aslında mesela bir denge tepkimesi şey yaptığımızda da, yazdığımızda da iki tane hız şeyimiz var, okumuz var. Yani olaylar reversible ise iki yönde de gerçekleşiyor aslında. Dolayısıyla mikro düzeyde baktığında evet, yani DNA ile DNA polimeraz birleşiyorlar belli bir hızda ama belli bir hızda da ayrılıyorlar. E, Ama zaten termodinamik o ölçekte bir tanımlama yapmıyor. Termodinamiğin çalışması için bir coarse grain lazım. Yani ve yani birden fazla e, element olması lazım ki kaotik bir sistem olabilsin. Kompleks ve kaotik sistem olabilsin. Dolayısıyla evet, termodinamiğe mikro düzeyde aykırıymış gibi görünse de aslında aykırı değil. Çünkü termodinamik kapsamında değil. Bir de şu oluyor, işte sanki biz termodinamiğin ikinci yasasına aykırı bir şey yaşıyormuşuz gibi dünyada bir şeyimiz var, algımız var. E, Çünkü yani normalde böyle bir karmaşanın, böyle bir kompleksitenin gelişmesini beklemeyiz. Karmaşa demek doğru değil. E, Ama bu niye oluyor? Çünkü dünya fikse ettiği kadar düşük entropiye karşılık dışarıya da infrared fotonlar yardımıyla yüksek entropi basıyor. Yani aslında biz bir lokal vortex gibiyiz şeyin içerisinde. Bütün bu evrenin yok oluş süreci içerisinde. Bir lokal vortexiz, yani kendi içerisinde bir oluşmuş bir kompleks, bir fraktal bir yapıyız yani.
Çok teşekkür ederiz hocam cevaplarınız için. Sayın Arda Deniz Dokuzoğlu'na sunum için teşekkür ediyoruz ve kendisi adına Tema Vakfı'na bağış yaptığımızı belirtmek istiyoruz. Sertifikasını mail üzerinden kendisine göndereceğiz.
Teşekkür ederim.
Çok sağ olun hocam geldiğiniz için.
Davetiniz için teşekkürler.
Our sincere gratitude to Mr. Arda Deniz Dokuzoğlu for his presentation, and we are pleased to announce a donation to the Tema Foundation on his behalf. His certificate will be dispatched via email.
Değerli konuklarımız, bir sonraki oturum için 37 dakikalık bir ara verilecek. İkinci oturumumuz saat 13.30'da başlayacaktır. Ayrıca chat kısmında paylaştığınız formu doldurarak çekilişlerimize katılabilir. Katılım formlarınızda oturuma katıldığınızı belirtmek için yine chat kısmında ve aralarda paylaştığımız QR kodlardan ulaşabilirsiniz.
Dear guests, we will have a 45-minute break before the next session. The next session will start at 1:30. Additionally, you have the opportunity to participate in our raffles by completing the form shared in the chat. To indicate that in attendance for the session, you can access our participation forms through the chat or the QR codes shared during the breaks.
Değerli misafirlerimiz, şimdi Sayın Elif Asena Meriç'i astrobiyolojik yolculuk, DNA'nın dünyevi zaman kapsüllerinde kozmik yankılara yolculuğu konulu sunumunu yapmak üzere davet ediyoruz.
May I now invite Ms. Elif Asena Meriç to present her talk on astrobiological journey from DNA's early time capsules to cosmic echoes.
Hello, everyone. Sorry. I will share my screen now. Hello, everyone. Thank you for this kind invitation. And okay, so let's start. In this talk, we will walk through the buried time capsules, which is ancient DNA, to its cosmic echoes, and what we can learn from it, and how we can apply it to the astrobiological research. First, I want to give you an analogy, and I want to ask you this. Imagine that I give you a huge box full of mixed puzzle pieces. What would be the first thing you would do to start doing this puzzle? Okay, this puzzle pieces looks a bit weird because it's been created by an AI tool, but we will answer the question at the end of the presentation. In the meantime, you can think about the answer. What is astrobiology? Perhaps it is an unnecessary question for this conference, but let's say it anyway. Astrobiology is a multidisciplinary study of origins, distribution, evolution, both past and future of life. And the field itself is not confined to Earth, but we have no confirmed samples of life forms nurtured in places other than Earth yet.
Çok affedersiniz.
Evet, şu anda evet ilerletmiyorum çünkü. When we think about astrobiological questions, the list would include what is life, how life has started, is there life elsewhere, under what conditions life starts, and how do you adapt to extreme environments? Of course, when you think about searching life in other planets or solar systems, the first thing that comes to mind is extreme environments. Either it's a frozen planet, or a very hot planet, or an environment that doesn't have enough oxygen in the atmosphere, or no atmosphere at all. That's why extremophiles and tough organisms are sparking our interests. And I think we can understand why tardigrades are the rock stars of astrobiology in that sense, with their toughness and being able to withstand harsh environments of space. And in this sense, adaptation capability comes to mind, which is a core basis of evolution, which you already know, being able to adapt. And luckily, we have a lot of organisms that can live under extreme environments, extreme conditions to be able to tackle their know-how, metaphorically, to be able to understand their unique skills and biological mechanisms that allow them to thrive under challenging conditions. We said that the adaptation is key here to understand the limits of life. And we know the conditions of Earth for our day, and also we know the past. We know that once Earth was warmer, when there was no oxygen, there was less oxygen, it had glacial periods, it had cold times. So this allows us something great, actually. We can look at the past organisms and try to investigate how they have adapted to their own environment in their own time. And even better, we can see how through time it has changed depending on the environmental conditions. This is where I would like to talk about ancient DNA, or environmental DNA in other words. Perhaps you may know it from the work of Nobel laureates who won Nobel Prize in 2022, Svante Pääbo. In his work, he used ancient DNA analysis to sequence a region of mitochondrial DNA from a 40,000 years old 40,000 years old piece of bone for analyzing Neanderthal genome. And they have discovered that the most recent common ancestor of Neanderthals and Homo sapiens lived around 800,000 years ago, which sheds light to human evolution. But it was first realized in 1984, I think, if I remember right, at the University of California, that they can actually conduct ancient DNA from a museum specimen of quagga, which is an extinct zebra subspecies that you can see in the picture. And this is the only specimen photographed. And they realized that they can retrieve ancient DNA from a specimen that died over 150 years ago. And through technological developments and research, now we can analyze not just museum specimens or human remains, but also plants and microorganisms. Even though 150-year quagga might be impressive at the time, now the oldest environmental DNA is 2 million years old. And it's an ecosystem. I'm not talking about a single sample here. Now we can investigate a whole ecosystem that was thriving 2 million years ago. So the DNA molecules are electrically charged, and some of the sediment particles in the soil are also electrically charged. And DNA will bind to these sediment particles, and this will reduce the degradation rate of the DNA. And if it's colder, that's even better, because the degradation will slow down even more. That's why the best place to look for ancient DNA is a permafrost, and permafrost is frozen soil. And that's where they took this 2 million years old sample. They've collected samples from Greenland in an area named Cape Copenhagen Formation. I hope I read it correctly. And at that time, DNA was thought to survive maximum 1 million years. That's why it was a slight chance for them to retrieve DNA from 2 millions. But the team went back to the area many times and tried again as the technology improved. And they were able to extract more short fragments of DNA bound to the sediments in time. And this is the area that they have collected the samples. And they were able to find animal DNA from 2 million years old. Like I said, the conditions of this place was perfect for DNA to be preserved, because there was permafrost, it was cold, and the remains have been protected really well due to this reason. And they were able to retrieve DNA from reindeers, hare, and mastodons. And mastodon is especially interesting because mastodons found in North America, but Greenland, where the sampling has been processed, at the time it was an island, so they would have to either swim there or perhaps they could walk over the sea ice. So this add something new to
Can
our
I
knowledge.
ask you something? I'm sorry I interrupted, but we are still on the beginning page on the screen. We
Oh,
were wondering if you were changing it and we don't see it.
yes, I am changing it actually.
Yeah, we don't see it, so
Okay, I will stop the presentation. Let me try to share again. Which slide can you see?
yeah, we can see it now. Thank you.
Görebiliyor musunuz? Anladım. Değiştiriyorum ama o zaman full ekran yapmayayım. Sanırım şu anda değiştirdiğimi görebiliyorsunuz. Bir önceki slaytlar o zaman tekrar baştan mı başlayayım yoksa hızlıca şöyle bir geçeyim? Çok hızlı bir şekilde geçeyim olmadı.
Evet hocam, uygun olur.
Evet. Bir rockstar tardigradımız vardı. And this is the quagga that went extinct. And this is the area where they have collected the samples for the 2 million years old environmental DNA. Okay, so we were here. They have collected, they have found animal DNA from 2 million years old, and the condition was perfect. And they have found, like I said, deer, hare, and mastodons. And it is important for our knowledge to understand the distribution of animals. And not just animals, but we can also include plants to the ecosystem and see a broader view and the interaction of the species. We can we can compare the ancient samples with their relatives from our current day and their previous versions and see the evolution through time. And this is an artistic impression of the same area, this area, and 2 million years ago, which is quite nice. Okay, what about microbes? The oldest evidence of life on Earth so far is these structures found in Australia. They are 3.48 million years old, called stromatolites. I think many of you know this already. And they are preserved remains of microbial mats stacked on top one another. But I will not talk more about it because it is Dr. Balja's area, and I'm sure she will give more details in her talk. But I want to talk about microorganisms here. There are some challenges of analyzing the ancient microorganisms: preservation conditions, DNA degradation, low DNA yields, lack of reference, authentication, ethical considerations, and sample contamination. But I think the most challenging is the contamination. That's one of the reasons that paleomicrobiology has developed rather later than other paleo fields, because it's been thought that the microorganisms that they found in ancient samples were just modern day microbes, because they could not be dormant. I mean, they could not be alive. But later it's been realized that these are actually ancient. And there need to be an extra caution while collecting these samples to avoid contamination. And of course, DNA work should be performed in clean rooms, ancient DNA work. and with special clothing and extra care. And microbial ancient DNA can be isolated from amber, from bones, dental samples, but perhaps the constant low thermal energy and water availability in permafrost makes it a better spot. These conditions can also lead the microorganism to dormancy. And one example for that is a eukaryote infecting virus, isolated, which is 30,000 years old, and it was still in a dormant state. Okay, we talked a lot about ancient DNA, but I should also say that when you retrieve ancient DNA samples, it will not be a perfect DNA strand. Sorry. Due to being preserved for a very long time, it also depends on the preservation conditions, of course. It will be degraded, which means that both strands will be broken, as you can see here. And it will be most likely be in short fragments. So let's say that you have some permafrost samples in your hands, not in your hands because it would contaminate, but after taking the sample, which is usually drilling blocks of frozen soil, and you need to take from the middle of this sample to avoid contamination from the outside as well. It will be taken to the ancient DNA lab within a sterile environment under a UV light, as you can see, and you need to wear multiple pieces of clean suits, double gloves, and after all these precautions, you analyze the DNA and make a DNA library. That's great. That's the wet lab part. After the wet lab part, you need to use bioinformatics to retrieve information, especially if you are working with microorganisms, because it might be and it will likely be unculturable. And since you can culture it, you need to use tools to analyze the DNA. And there are many tools that's been developed by amazing teams out there already. Without the details, with these tools, what you can do in general is that you can, since it is you just collect the soil, so it's a mix of everything, right? So you have plants, animals, microorganisms, everything is mixed. But with these tools, you can separate the eukaryote and prokaryote DNA, and you can also, as I said earlier, that DNA is degraded, it is in short fragments. You can take these short fragments and assemble them into longer sequences, which is called contigs. And with these longer sequences, you can even use tools to make a complete genome, which is called binning in this step. And it will, of course, contain a certain percentage of completeness and also contamination. But this is what we do now. This is also what I am doing for my master's thesis. I'm working with ancient microbial DNA, and I'm analyzing it for certain secondary metabolites. Well, so how is it all related to astrobiology? We talked a lot about ancient DNA. Do you remember the question I asked in the beginning? What would be the first thing you do if I give you a pile of puzzle pieces that is mixed? Well, you would look at the picture on the box, because without knowing the picture, you can't put the pieces together. Maybe you can, but it will take a lot longer, and especially if the puzzle is enormous, not just this 500 pieces puzzle, of course. And if you have missing pieces in this puzzle, you may not realize it without having the picture. Just like that, in astrobiology, to be able to look at the stars and planets and search for life elsewhere, you need to understand the life on Earth. Earth is also pretty amazing and interesting planet as well. And we have so much diversity and a lot to discover from present and also past. And ancient DNA is, for now, it seems like the best option to understand the genetic material, DNA, and its adaptation, its change through time. And we will probably advance our technology for ancient DNA methodologies. And perhaps when the day comes, we might even try to analyze the ice caps of Mars for possible ancient DNA remains. Maybe they already have plans or projects for it. And yeah, what if it had a DNA? But I don't know if it's a DNA or not, which is a different discussion. But thank you for listening. And I would like to thank the organizing committee for inviting me, especially to Simay. Unfortunately, I won't be able to take questions, but you can always reach out to me on social media. This is my Twitter account. You can write about anything. And thank you again for inviting me, and I hope you all have an amazing conference.
Sayın Elif Hasan Meriç'e konuşması için teşekkür ediyoruz ve kendisi adına Tema Vakfı'na bağış yaptığımızı da belirtmek istiyoruz. Sertifikasını mail üzerinden kendisine göndereceğiz.
We extend our appreciation to Ms. Elif Hasina Meriç for her presentation and are pleased to convey our donation to the Tema Foundation on her behalf. Her certificate will be sent electronically.
Değerli konuklarımız, bir sonraki oturum için yaklaşık 1 saatlik bir ara verilmiştir. 3. oturumumuz saat 15.00'da başlayacaktır. Ayrıca chat kısmında paylaştığımız formu doldurarak çekilişlerimize katılabilir. Katılım formlarımıza da oturuma katıldığınızı belirtmek için yeni chat kısmında ve aralarda paylaştığımız QR kodlardan ulaşabilirsiniz.
Esteemed guests, we have a 1-hour break ahead of us. You may also participate in our raffles by completing the form shared in the chat, and attendance forms are accessible through the QR codes and links provided in the chat and during breaks.
Değerli misafirlerimiz, şimdi Sayın Nurgül Çelik Balcı'yı neden erken dünyayı önemsiyoruz, evrendeki yaşam arayışı için ne öğrenebiliriz konulu sunumunu yapmak üzere davet ediyoruz.
May I now invite Ms. Nurgül Çelik Balcı to present her talk on why do we value early Earth? What can we learn from the search of life in the universe?
Ekranı paylaşacağım. Değil mi? Okay. Okay, share. Biraz müsaade edeceksiniz. Onu anlamaya çalışıyorum oraya. Okay. Well, a pleasure to be here, although I have some head problem, but I'll do my best. Thanks for the invitation. Today I'm trying to explain you from a geological perspective, why do we really care about early Earth? And what can we learn in the search for life in the universe? Why do we need to know our own past? So what can we learn from this past for the entire cosmos, in fact? Well, I would like to take a minute and try to explain for those who may be interested in such studies. So we do have a geomicrobiology and biogeochemistry members. And so we were really privileged to study many of the international known people, such as Bruni from University, Tanya from MIT, Alexander from University of Cambridge, and many really well-known scientists in our lab with our groups. And so let me know if you are interested in getting involved in this kind of research. Here is our link and my email for further questions and anything. Not necessarily, it doesn't have to be related to this talk, but you can also, reach me. So from this, I would like to start with geological time scale. So for this talk, I was, it's astrobiology, and it mostly related to the biology. But so I'm trying to make a sense for you that we do really need to know geology. So I'm going to start with what is early Earth? What do I mean when I say early Earth? So this is a geological time scale. Most of our geology students are really familiar with this time scale. And so we're talking about 4.6 billion years almost, right? So we have different times. We call it a different eon, era, period, and epoch. But today I'm going to mostly talk about the early Earth, which is it's shown with this we call Precambrian, for umbrella term Precambrian. But when we talk about the early Earth, we talk about mostly Archean and Hadean and Proterozoic. What I like to emphasize in here that most of the Earth history happened during the Precambrian, which is early Earth. So think about it as a whole, the Earth, if you take the kind of graphical presentation saying that Proterozoic making up 42% of Earth history, Archean 33%, and Hadean it's a 13%. So Phanerozoic time, it's almost 12%. It's only made up small portion of the Earth history, which the complex microorganism evolved. So if you try to understand from this perspective of Earth history, that we do really need to know the early Earth history, try to make sense of how life evolved just by looking at this geological time scale. So it's almost 88% of our history belongs to early Earth time. So what about if you take the evolution of life on a 24-hour scale, and you can see unlike common belief, Earth, it's 88%, almost 90% of its history was really habitable. It was really life-friendly environment we're talking about. See, you started life at around 3.8 billion years ago, and mostly was phototrophic microorganisms, and then evolved into the first eukaryotic cells around 2 billion years ago, and then it started to evolve into more complex life. But when you look at the early Earth time, we're talking about the really microbial life, and Earth was in habitable most of the time. So early Earth, we kind of have idea in the geological time scale what is early Earth, and when you look at the 24 hours of the life on Earth, then you may have some kind of a feeling, why do we really need to know the early Earth? So when we talk about the early Earth, it's actually we're talking about trying to understand the alternative Earths where life might exist. So we know that the Earth that we live in right now, it wasn't like this. It's constantly evolved. Then every different stage that Earth exists may have life in a different way. So geochemical environments, so environments of evolving Earth was different, but life was always there. So by looking at this alternative history, or we call alternative Earths, then we can really get lesson from this early Earth condition, where to go for searching life in the universe, it's basically either in our solar system or beside, out of our solar system. We need to know the alternative Earths. So right now, since astrobiology, I like to talk about a little bit with top candidates right now. Okay, this is constantly changing, but now I think there is a huge consensus about the top candidates for the search for life. For example, Europa, it's the icy moon of Jupiter, as you know, and has a liquid ocean beneath the ice sheet. The surface indicate that some chemically interesting is going on beneath the ice. So it's one of the #1 candidates. Enceladus, like Europa, Saturn moons has liquid water, and is shown by the Cassini space probe. And Cassini detected methane from Enceladus, but methane is it's not necessarily always the biological compound. There's a non-biological processes can also produce methane, but still exciting news. There's like different organic compound, carbon compounds that in the different planetary symbols has. So methane is considered as the possible biomarker, but still there's a question mark. Venus, we all know about the Venus. It's recently with the phosphine. So phosphine is really it's a biomarker on Venus, but we still have, there are purely chemical reaction that may produce phosphine, but you still needed some kind of biology, as you know, from Earth to produce these compounds. Mars, there's no need to talk about Mars, and since 1970s, even earlier, there has been really good candidate for life, search for life in the universe. And Titan is really interesting. It's a Saturn moon. But when we talk about the Titan, we are going to highly speculative because Titan is so cold. Even though the lakes and liquid methane it exists, but life on Titan would require a different biochemistry we may not familiar from the Earth. So even when we think about it, so how are we going to recognize life on Titan is really the challenging question. But still, these are the top candidates right now that we're really looking for, searching for life in the universe. So when we talk about these top candidates, I would like to relate it to this, the early Earth conditions. So which one of these is really resemble the early Earth conditions? Is it Mars or Venus, or maybe resemble in saying that which alternative Earths could be very similar to one of these candidates. So that's important. So when we talk about the Earth and early Earth history, so present and 2.5 billion years and 4.2 or even 4.6 billion years, we're talking about a totally different type of Earth. So Earth has 4.2 billion years history of liquid water, which is, we know, the precondition for life. But through that time, oceans were forming, continents were emerging, temperature were rising and falling, and the composition of the atmosphere and ocean was constantly changing. So if you can think of these varying conditions from the planet past as alternative Earths, then by understanding is the long history of alternative Earths, we can increase the number of places or that is supposed to be or supporting the worlds, and try to understand the factors that defined or help to flourish life. So that's why early Earth conditions and understanding this is important. But what we want to do is it's really unreveal of our planet past by studying alternative version of the Earth. From this, mapping potential areas harboring in the universe based on the geological, chemical, and biological data of the continents, oceans, and atmosphere of the Earth's different time period. Try to understand our past. So from the lesson of the Earth, it's gonna take us through the universe in somewhere that life is likely possible. So to do that, we just need to know what was going on early in the Earth. So let's check it out when we talk about the early Earth, what was happening. So 4.6 years old Earth, 4.4 billion years ago, Earth core was already formed, crust started have formed, atmospheric gases was possible. And more importantly, we know that liquid water was available. At least there's a huge data and consensus about the liquid water was there. So how do we know all this stuff? So there are some studies and publishing and data from different part of the Earth that is give us really about the past. For example, we know that early crust, it's the Hadean mafic crust, for example, which is rich in iron and magnesium. So it's the mafic in composition in specific basaltic rocks. So we see these basaltic rocks in early crust, and we also see on the some part of the Mars, which is Jezero crater. So we can see these similarities, and you maybe even tell us that why these similarities exist, and if these similarities we see in the context, mineralogical context, may help us to define the really hot spots on Mars for life flourishing environment. So what about the early Earth atmosphere? We know that reducing atmospheric chemistry was available in the early Earth, for example, rich in water vapor, CO2, some nitrogen compounds, and methane, and some of the sulfur compounds. So there's energy source was available as well. For example, UV radiation and volcanic eruptions. It was providing kind of flourishing environment for life. So there's energy available, and there's some hydrogen, carbon, nitrogen, and sulfur, which is the building block of life, is available. And plus heavy meteor bombardment, it's even making ideal, favorable conditions for life. So, and luckily there is no free oxygen yet. The organic molecules preserve once form. So with the early Earth, yes, it's providing nice environment for the origin of life, but there are also some the unexpected or negative effects of early Earth is the cosmic bombardment. So it was keep continuously meteor and asteroid hitting the early Earth. That kind of it sounds like an uninhabitable environment, but in fact, the recent studies show that this cosmic bombardment even may create a nice environment. For example, the impact history of Earth. This is the 2021 publication that came out recently, that the impact history of Earth suggests that the impact called Moneto occurred 4.2 billion years ago, in fact, may create favorable conditions for RNA or precursor RNA molecules and even preserve them. So this is the fairly new ideas. Even this lately impact may create atmospheric conditions that is necessary to produce the RNA molecules that require really nitrogen-rich compounds. But there is also the bad side of this cosmic bombardment. It's just raised very early rock records, so which leave us empty-handed. But luckily, there are some early rock records preserved on Earth. So you see the map now that's showing the distribution of Archean rocks over the Earth. So there are some famously known Pilbara section on Australia that is preserved Archean rocks, and also some places in Africa and the United States and the South America. There are some Archean rocks that people have been working for a long time, and it's continue to working to get history of life. So yes, the early atmosphere condition is favorable for life, and there's some impact that recently the new theories, one of the new theories for origin of life was occurring. How do we know what is the signals of life or imprint of life that preserve in the rock record? So this is one of the examples that we use as a proxy, the carbon isotopes. The carbon isotopes is two different carbon isotopes. 12 and 13 has different neutron numbers, and as a result of the different weight, so they react or act different in a geochemical conditions. If there's biology, they act differently. If abiotic reaction, they have differently. But using this chemical behavior of carbon isotopes, people was able to create the inorganic carbon versus organic carbon composition. So as you can see from here, the carbon 13 values change over the history of Earth. So as you can see, the inorganic carbon has different values versus organic carbon. By using these differences, we know that the life preserves some of the carbon isotopes. This is one way to read the signal of life recorded in the rock. So, for example, carbon-depleted carbon microparticles from the Greenland was the first, one of first robust evidence for life. And later on, even older, the zircon samples, zircon kind of mineral, the biogenic carbon preserving the zircon from 4.1 billion year potentially suggested the biology. So this is one way to read early rock records. The second one is the sulfur isotopes. Then you can see the sulfur isotope throughout the Earth history. That is, the blue one is belong to pyrite type of mineral, and the red one is sulfate. So you can see this throughout the time, these values are changed. And there's a sulfate reduction, mostly biological reactions. And we certainly know that after the oxygen is accumulating, that for 2.5 billion years ago, and start accumulate in the atmosphere and the ocean. And as a result of this accumulation, pyrite oxides produce sulfate, and sulfate reduction which biologically leave their fingerprints on the rock. So it's the sulfur isotopes that we can use in the rock record to track down the biology. So what about the other bioessential elements? For example, nickel, cobalt, and zinc, which we know that these are essential transition metals that is required to metabolize, required to produce proteins and as a result, the genes. So by looking at the changing ocean chemistry and redox condition throughout the time, we know that these elements and concentration of these elements change. So by looking at this profile of these elements, that we can differentiate the geochemical records, life records on the rocks. For example, when you look at the very early time on Earth, for example, 4 billion years ago, you can see in the ocean iron rich, manganese rich, cobalt rich, and nickel rich. But when your time came to Paleozoic, where all the complex life evolved, then we can see differences in ocean chemistry and redox condition. For example, low in iron, low in manganese, cobalt, and nickel. These are the good indication of changing ocean chemistry as a result of changing geochemical conditions that life adapted. So as a result of reading the geological records, that we were able to see the milestone of life. So when you look at the milestone of life, we know that life, the first record on rock record is around 3.8 billion years ago, which is the preserving stromatolites, and came as the photosynthetic microorganisms. After that, you can see 2.7 billion years ago, cyanobacteria dominated. And then after that, it's around 2.5 billion years ago, the oxidation events happened, and the oxygen started to accumulate in the atmosphere following the oxygen, following the photosynthesis reaction. And life started to evolve into more complex life forms around like 0.6 billion years ago. So this is the milestone of life, but we usually, if you're interested in the early Earth and the habitable environments, then we actually interested in mostly 3.2 and 2.5 billion years ago, when the earliest form of life began to release oxygen into the atmosphere. Then you can see around here, and we have photosynthesis reaction, and then these great oxidation events flooded Earth atmosphere and ocean with oxygen. Okay, it's actually paved the way for complex life. So how do we know all this? By just reading the rock records. So even the chemical footprints of life in the rock, the banded iron formation. So following great oxidation events, even before, way before than that, iron, the ocean was lack of oxygen. So iron came in, released into the ocean via volcanic activity, was in the form of iron 2. But there are some photosynthetic microorganism making oxygen was able to produce iron 3, which result in iron oxide. And this is the result of this biological activity. So this part of the rock we call banded iron formations, you know, person that took my classes in geochemistry classes should know this banded iron formation, that we just all the time talk about these formations. So this is red iron oxide is a good life record. So the second life record is stromatolites, the oldest life forms. So you can see one of this Varavona group, the 3.5 billion years old, and the sulfur isotope data nicely show biological activity. The one that is in there's a very small storm that you see on the slide is from Greenland. It's supposed to be the 3.7 billion years old stromatolites, but which is published in the Nature. But they came up later that this was actually it was an artifact. So there's still debate on if this really represent the life form or not. So stromatolite, it's known very common in the Proterozoic time, and then which is a sedimentary structure that formed by microbial communities. So it's very common in the Proterozoic time, but it started declining their diversity. It's throughout the Phanerozoic time. It's about 700 million years ago. So which is, although it's not certain, the decline in this diversity is really related to the to more appearance of material zones, but still not quite sure why we don't see why they are not as common as earlier. But so these are the fossil stromatolites, Precambrian stromatolites, which is 3.4 billion years old. And there's also different type of stromatolites. So you can see the differences in terms of morphology. But we don't really know this morphology is really a result of biogenic versus abiogenic activity. But some of them, we can see there are some certain reactions or condition they may produce stromatolite-like structure. So these are more examples from different places. For example, this is from Canada, and this is from South Africa that is preserved nicely. And this is from Lake Salda, that is the recent one. They produce the microbial production of stromatolites. So another thing is that we need to think about the analog places on Earth, either on early, or, either in they form very early in the rock records, or it's the modern times that we need to study these places so that we can get a perspective for our perspective where to look for life. So I'm sure all you kind of at least familiar with the Lake Salda and why we study here. And there are some carbonate, magnesium carbonate, which is has some similarities with those identified in Jezero Crater. We're currently working in this section so that we can try to understand the signature, biosignature, or the life signals that preserve in carbonates. So there are even more places. For example, Hamillion Pool in Shark Bay, Australia, that's still there's stromatolites, or in more general microbialites that are forming in that environment. The people are, one of our colleagues, Tanya, it's working in this section, in this part of the area. So there are fossil stromatolites and biosignature. We know that stromatolites produced by biological activity. So we know that they can produce biological activity, but what if this, or what extent this microbialites actually preserve these biological signals? Then we need really both fossil and current stromatolite to compare them and just see if there's any difference in terms of really identify life records. So we also working with the fossil stromatolites in Lake Salda. It's fairly young, but still considered fossil. So one thing is I like to emphasize is that even though there are some fossil microfossils, which is I show here from Warrawoona prokaryotic microfossil, 3.5 billion years. It's one of the best example of fossil record. But there are some studies that I'm showing on the slide that they're purely abiotic processes. They produce very similar cyanobacteria type structure. So we call biogenicity problem. And I would like to give you the feeling that how this searching for life in the universe is itself, it's difficult. Even though you might have a technical novel instruments and everything, but sometimes it's really hard to recognize life signals. So the other one is that we need to think about the atmospheric chemistry. So I'm sure we all agree in a way that searching for life in the universe, it's challenging, and the best way to do is looking for the atmosphere, because you can monitor the atmosphere from anywhere. So if that's the case, we need to concentrate in atmospheric chemistry. And if you think about our own planet, and if you've seen our planet from the distance, you see this blue-green color. Green means life. So we can detect how far it is. It doesn't really matter. So that's give you an idea that atmospheric chemistry would be the nice target for searching for life. And this is the pictures from Australia. I'm trying to remember the lake that is full of extremophile. In this case, haloarchaea. As you can see that they produce different pigments. This pigment, different pigments, and in this case, rhodopsin pigments. If we can determine the different pigment color, might result from photosynthesis under different stellar irradiation or UV, and if they produce different biogenic gases with different color, we actually use this photosynthetic biosignature. So what I'm saying is that we need to be very open-minded and looking for different color in the atmosphere if possible, as a biosynthetic signature. But there are some really attractive false terminology that we need to consider when we're looking for the search for life. For example, false positives we define as consider as trace of life, but without life traces. For example, oxygen is not necessarily always the result of life. But the other one is false negative, which means occur as a result of life effects, but escape our detection and make us consider that there is no life. This is a false negatives. And I'll show you, for example, these are the one of the examples that give us both false negative and false positive. For example, methane and oxygen, they're here and evaluated throughout the Earth history. So you can see the very early Earth time, during the Archean time, and oxygen and ozone wasn't detectable in the atmosphere, but methane, on the contrary, methane was in the atmosphere. Throughout the time, this changed. So we know that the presence of both methane and oxygen in the atmosphere has been viewed as a gold standard in the search for distant life. But these two gases should not coexist in appreciable amount because they react rapidly. But if there is living organisms can constantly replenish them into the atmosphere, then we really need to understand the equilibrium between methane and oxygen. Okay, so if you can understand this equilibrium, we can relate it to life. But so finding methane in the atmosphere of any type of planets doesn't mean that there's life, and the oxygen as well. Okay, so we need to consider this these two biosignature, supposed biosignature, and try to understand the relationship between these two gases before making the jump to conclusion. So biosignature, we know that biological gas is oxygen buried in the oceans during the early Earth time before accumulating in the atmosphere. So what I'm saying is that when you travel to Archean time or the very early Archean time, there's no oxygen in the atmosphere, but there's oxygen in the ocean and there's life. But could be vice versa. The oxygen could be in the atmosphere, but not necessarily always biological. There's some For example, CO2 photolysis can produce oxygen. So if you have nitrogen, CO2, the water reach atmosphere, abiotic oxygen can be produced as well. So this is really getting more and more complicated, but if you know what you're dealing with, and if you get the lesson from early Earth as well, then you can make really sense out of all these obstacles. So the strongest evidence in the search for life in the cosmos, it can be very provided by photosynthetic biosphere. So if we study the atmosphere of any planetary planets, could be a very useful. But looking at oxygen, ozone, methane, some of the organic compounds, nitrogen-rich compounds would be a really good target. But we need to be careful about the false positives and try to relate it to other proxies. For example, as earlier suggested, iron, and also methane, iron, nickel, zinc, all these geochemical proxies should be used together. So I think I'm gonna finish up with this last slide, and thank you for the invitation. I hope that I'll give you some sense why do we need to study early Earth.
Çok teşekkür ederiz hocam. Birkaç tane sorumuz var.
Okay.
İlk soru şu. Jeolojik olarak önemli reaksiyonlar dediğimiz zaman karşımıza ne tür reaksiyonlar çıkar? Reaksiyonlarda kullanılan maddeler bize o zamanki dünya koşulları hakkında nasıl bilgiler verebilir?
Bence güzel bir soru. Geniş de bir soru. Özellikle jeolojik silikatlar dediğimiz kayaçların karbondioksitlerle birlikte ayrışması aslında bizim burada olmamızın temel nedenlerinden bir tanesi. Düşünün, karbondioksit atmosferde ne kadar yüksekse o zamanki okyanusların asitik olma ihtimali çok daha yüksek. Ve sıcaklık da bu reaksiyonlarla birlikte değişiyor. Yani jeolojik kayaçlar aslında bir bakıma yerkürenin termostatı gibi davranmışlar. Birçok farklı reaksiyon gelişmiş. Biz şimdi, peki bunu nereden biliyoruz? Bu güncel zamanlarda hazırladığımız deneysel çalışmalarla bunlar aslında verifikasyona tabi tutuyoruz. Güncel koşulları çalışarak, güncel koşullardaki kayaç ayrışmalarında çalışarak geçmiş dönemler içerisinde neler olabileceği hakkında veriler elde ediyoruz. Umarım cevaplayabilmişimdir. Geniş, gerçekten güzel bir soru ve bence kapsamlı bir, daha kapsamlı bir araştırma gerekir diye düşünüyorum.
İkinci sorumuz şu. Elimizde bir örnek olsa, örneğin kaya parçası, bunun özel bir örnek olduğunu, yaşamın başlangıcı için bir şeyler ifade ettiğini nasıl anlarız? Jeolojik ipuçları ararken nelere dikkat ederiz?
Şimdi öncelikle bir kayacın yaşını anlamak lazım, değil mi? Güncel, yani milyon yaşında bir kayaçsa başka şey için kullanabiliriz ama yaşamın başlangıcına dair ya da o döneme ait bir kayaç olmadığı için o dönemdeki jeolojik koşulları anlamamız çok zor olur. Farklı sorular için cevaplayabiliriz. Mesela Avustralya'ya gidip Bandalar Information'dan bir örnek alabilirsek süper olur. Bunu elde edeceğimiz, okuyabileceğimiz bütün jeokimyasal veriler bize o dönem içerisinde hem atmosferi hakkında hem okyanus hakkında önemli ipuçları sağlayabilir. Demek ki öncelikle o kayacı bir yaşlandırmamız lazım. Ondan sonra da kayacın bileşimlerine bakmamız lazım. İçinde bulunduğu dönem içerisinde birçok veri yorumlamaya açık olabilir. Ama tek başına bir kayaç üzerine birçok analiz yapmak lazım. Birçok farklı veriyi kullanmak lazım. Hem benim size gösterdiğim bir izotop verisini kullanmak lazım. Hem kayacın yapısını kullanmak lazım. Hem kayacın içerisindeki mikrofosil var mı? Varsa bu yapıyı koruyabiliyor mu? Onu bile bulsak, size gösterdiğim gibi bu mikrofosillere benzeyen başka fosiller de üretilebiliyor laboratuvarda tamamen. Hatta organik bileşik bile bulmak aslında orada canlının olduğu hakkında bize bir fikir vermiyor. Çünkü birçok tamamen kimyasal koşullarda da organik bileşikler oluşturulabiliyor.
Çok teşekkürler. Şimdi üçüncü sorumuz şu. Son dönemdeki yaşamın kökenini anlama çalışmalarında Türkiye'den çok bahsediliyor. Siz bu durumu nasıl değerlendirirsiniz?
Türkiye'den bahsediliyor derken?
Haberlerde olsun, işte araştırmalarda olsun.
Yani şimdi tabii özellikle Salda Gölü'nde yapılan çalışmalarla birlikte popüler bilimde de çok etkin olmaya başladı. Böyle olunca işte Mars'a giden Perseverance robotuyla birlikte, gezginiyle de birlikte insanlar çok daha fazla bununla ilgilenmeye başladı. Ve verilere ulaşmak, dinlemek, bütün bunları bence hızlandıran aslında katalizör olan süreçler oldu. Tabii sağda yönde yaptığımız çalışmalar, orada NASA'nın parçası olarak devam etmek bence gençler üzerine çok önemli bir etki yarattı. Bu da bu tür çalışmaları ivmelendirdi diye düşünüyorum. Türkiye'nin yavaş yavaş başlayan uzayla ilgili çalışmaları da eminim buna katkıda bulunuyordur.
Çok teşekkürler. Son sorumuz şu. Dünyanın yaşı ve yaşamın başlangıcı arasında uzun bir zaman dilimi mevcut. Bir yaşamın oluşması için uygun koşullar sağlandığında bu süre kısaltılabilir mi? Bununla ilgili yapılan çalışmalar var mı?
Yani şu anda yaşamı yerkürede, en azından yerkürede, yani yerküre bildiğimiz yaşam barındıran tek gezegen. Yerkürede yaşam nasıl başladı? Bu verdiğimiz rakamlar bile şu andaki elimizdeki bilgilerle elde edilen veriler. Bazıları çok daha farklı rakamlar da söyleyebiliyor yeni verilerle, yeni çalışmalarla. Bu soruya cevaplayabilmek için zor bir soru. Bu soruyu cevaplayabilmek için öncelikle galiba yaşamın nasıl başladığını anlamamız lazım. Hangi koşullar gerekiyordu onu bile bilmiyoruz. Şu anda çok değişik teoriler var. Mesela artık impact kraterlerle yaşamın başladığı düşünülüyor. Belirli dönemlerde impact kraterin yaşamı yok ettiğini düşünürken şimdi impact kraterlerinin yaşam için gerekli olduğunu düşünüyoruz. Hatta RNA dünyası, RNA world dediğimiz yeni teorilerle birlikte Origin of Life'ın oluşma koşulları da değişti. Bu yüzden gerçekten şu andaki bilgilerle buna cevap verebilecek hiçbir fikrim yok. Bence önce yaşamın nasıl başladığını anlamamız gerekiyor galiba. Ondan sonra bunu kısaltabilir miyiz? Hiçbir fikrim yok o konuda. Olabilir diye düşünmek isterim.
Çok teşekkür ederiz hocam hem cevaplarınız için hem sunumunuz için.
Ben rica ederim. Teşekkür ederim tekrar davet için. Umarım başarıyla devam edersiniz. Görüşmek üzere. Kendinize iyi bakın.
Sayın Nurgül Çelik-Balcı'ya konuşması için teşekkür ediyoruz ve kendisi adına Tema Vakfı'na bağış yaptığımızı belirtmek istiyoruz. Sertifikasını mail üzerinden kendisine göndereceğiz.
Çok teşekkür ederim. Görüşmek üzere. Hoşçakalın.
Our gratitude to Ms. Nurgül Çelik-Balcı for her presentation, and we are pleased to announce a donation to the Tema Foundation on her behalf. Her certificate will be dispatched via email.
Tamam galiba değil mi arkadaşlar?
Teşekkürler
Görüşmek üzere.
hocam.
Hoşçakalın.
Görüşmek üzere.
Arkadaşlar şimdi Kaot'la günü kapatıyoruz. 5 dakika içinde yarışma başlıyor. Arkadaşlar 1 dakika süre verdik her soru için. Çünkü yayın 20-30 saniyelik bir gecikmeyle geliyor. Soruyu hemen göremezseniz panik yapmayın. Bir süre sonra göreceksiniz, o şekilde cevaplarsınız. Muhtemelen 10 kişiye tamamlanır. Son bir kişi kaldı. Başlatıyorum. Hepinize bol şans. İlk sorumuz geldi. Evet, sonuçlar geliyor. Sadece bir kişi doğru bilebilmiş. Kim olduğunu görelim. İsminde 2 tane emoji olan arkadaşımız birinci şu an. İkinci sorumuz geliyor. Titan hangi gezegenin uydusudur? İkinci sorumuz. Bu soruya 4 arkadaşımız doğru yanıt verdi. 4 arkadaşımız Venüs diyerek yanılmış. Ortaokul fen derslerinden aşina olduğumuz bir şeydi. Üçüncü sorumuz geliyor. Son 15 saniye bakalım. Scoreboard'da değişiklik olacak mı? Toplam 7 soru var arkadaşlar. Beş kişi doğru bilmiş. Çok iyi bir sayı. 3 arkadaşımız yanılmış. B birinci sıraya yükseldi. N çok az bir puan farkıyla onu takip ediyor. Dördüncü sorumuz geldi. Zor bir soru. Bakalım. Yine 5 arkadaşımız doğru yanıt vermiş. İpuçları işe yaramış gibi görünüyor. 4 kişi yanılmış bu soruda. Birincilik değişmedi. Ve yine 3 kişi zirveyi takipte. 5. sorumuz geliyor. Yine zor bir soru. Ama Instagram hesabımızda değindiğimiz konulardan biriydi. Bakalım yine skorboard değiştirebilecek nitelikte bir soru. Evet. İlk kez çoğunluk yanıldı ve 3 kişi konservat diyerek puanı kazandı. Bakalım B yine birinci. Ve ök ve yeşil sıvılı deney tüpü onu sıkı takipte. Altıncı sorumuz geliyor. Yaşamın kaplıcalarda başladığını düşünen bilim insanı. Yine kaplıcalar teorisinden de bahsetmiştik Instagram hesabımızda. Şıklar sizi yanıltmasın. Son 10 saniye. Geri sayıma başladık. Bakalım kitabı kim kazanacak sponsorumuzdan. 4 kişi David Dimmer diyerek bu soruyu doğru cevaplamış. 4 kişi de yanılmış. Bakalım zirvede değişim var mı? B yine doğru cevaplamış. B çok inatçı kitabı kazanma konusunda. Ök ve deney tüpü yine sıkı takipteler. Onlar da inat ettiler. Şimdi gelelim son sorumuza. Artık zirveye giden yolda son çıkış yolu bu. Yine yanıltıcı şıklar var. Bakalım kitaba giden yolda B zirvedeki inatçılığını sürdürebilecek mi? Ök ve deney tüpü bu sefer onu geçebilecek mi? Son 5 saniye. Ve 4 kişi bu soruyu doğru cevaplamış. 3 kişi yanlış cevaplamış. Bakalım. Kazanan geliyor. 3. deney tüpü olmuş. Maalesef tutunamamış zirveye. 2. ök olmuş. Evet. B. B geçilememiş. Gerçekten çok inatçıydı. Yarışmanın başından beri sıkı sıkıya tutunduğu koltuğa bırakmak istemedi. İnat etti ve başardı. B eğer canlı yayındaki sohbet kısmına adını soyadını yazarsan ve YTÜ Biyogen hesabıyla iletişime geçersen ödülünü alacaksın. Ben tebrik ederim seni. Yarışan herkesi tebrik ederim. Önemli olan burada eğlenmekti.
Evet. Eğlenip bilgilenmek.
Ben bilgilendiğimiz kanaatindeyim bugün. Yıldız Teknik Üniversitesi Biyoteknoloji ve Genetik Kulübü olarak bu sene 3.'sünü düzenlediğimiz Astrobioloji Konferansi etkinliğimizin ilk günü sona ermiştir. Bizleri onurlandırarak davetimizi kabul eden bütün konuşmacılarımıza ve siz katılımcılarımıza teşekkür ederiz. Katılım belgeniz için birazdan paylaşacağımız linkten 3. oturumunuzun katılımını sağlayabilirsiniz. Gelecek etkinliklerimizden haberdar olmak için sosyal medya hesaplarımızı takip edebilirsiniz. Yarın etkinliğimizin 2. gününde görüşmek dileğiyle. Sağlıkla kalın, bilimle kalın.
The first day of the Astrobiology Conference, hosted by the Biotechnology and Genetics Club of Yıldız Technical University, has reached its conclusion. We extend our thanks to all our esteemed speakers and participants. You may access the third session for your attendance certificate through the link to be shared shortly. Stay informed about future events by following our social media accounts. We eagerly anticipate your presence on the second day of our event. Stay healthy, stay scientific.